So, as I said, of the number of subtypes that a CD4 positive mature cell can develop, the regulatory subtype was an important subtype. Then in the last slide, I just very much hastened this part, that this is basically a life cycle of a T lymphocyte. We have seen the development of a T lymphocyte. So when the T lymphocyte develops and finally matures and come out of a thymocyte, what happens? If in its lifetime, it meets an APC and interacts with a peptide MHC complex via its T cell receptor, then only its activation is accomplished and the T lymphocyte functions, divides, produces a memory cell, and once that antigenic load is removed from the body, this activated T lymphocytes is also signaled to get inactivated. So this is basically the antigenic path of a T cell. So antigen uptake, antigen presentation by the MHC class 2 or 1, then downstream signaling events, activation of the T cell, and once that antigenic load is gradually removed from the blood or the peripheral lymphoid organs, further inactivation of the T lymphocyte organs. This is a picture of licensed APC. What does that mean? That means that an antigen presenting cell, consider an antigen presenting cell. Let's go back to an antigen presenting cell. This is an antigen presenting cell. An antigen presenting cell can express both MHC class 2 and MHC class 1. It was initially thought that both the lymphocytes can recognize MHC class 1 peptide or MHC class 2 peptide simultaneously, and the pathway can simultaneously proceed. But later on, this concept was challenged, and it was seen that an antigen presenting cell initially first interacts with a CD4 positive mature T lymphocytes. Only after the complete interaction and the signaling process is completed, then only the antigen presenting cell further, if it has any self antigens to express, further expresses the MHC class 1 with I mean to say with mutated self antigens, and express it to the T cytotoxic cells. So this model actually signifies the fact that for an antigen presenting cell to present antigens to cytotoxic T lymphocytes, it needs to be primed by a CD4 positive T lymphocytes first. It needs to have an interaction with the CD4 positive T lymphocytes first to become activated. And this kind of activated antigen presenting cells can later on also present antigens via their MHC class 1 to the cytotoxic T lymphocytes. But mind it, this phenomena happens in the peripheral blood or in the peripheral lymphoid tissue, not in the thymocytes. Within the thymocytes, the epithelial cells are capable of simultaneously presenting the MHC class 2 and MHC class 1 together. There is no first and second, but in case of the antigen presenting cells in various tissues, in the lymphatics, in the blood, it has been seen that the APCs need a priming by the mature CD4 positive cells first, and once they are activated or signaled by the mature CD4 positive cells, then they have the license to also present antigens via the MHC class 1 to the CD8 positive or the T cytotoxic cells. This is a picture of the central tolerance, that is the tolerance that we observed in the thymus. If there is a TCR rearrangement, there is a productive rearrangement. I'm not getting into what is productive rearrangement. My PG batch, students from my PG batch are now aware what is a productive rearrangement and what is unproductive rearrangement. But for fourth sem, let's stick to that at the DNA germline level, if the rearrangement is not proper, those cells are forced to die. If the germline DNA is properly rearranged for the alpha and the beta chain, we get the pre-T cell and Then see, there are three factors written: no avidity, low avidity, high avidity. Avidity is the total of the immune interaction between antigen presenting cell and T lymphocytes. So low avidity means moderate avidity. So the pre-T cell, T lymphocyte has moderate avidity for antigens. If it does not recognize any self-antigens, that is no avidity, then also apoptosis. If it gets hyperactivated by self-antigens, then there is neglect by death or clonal deletion. Or if there is no recognition by any antigens also, then also clonal deletion. However, the moderately activated T cells mature and finally give us a single positive CD4 and CD8 cell. This is a very schematic picture. This was a picture to just highlight central T tolerance, but for fourth semester, if you don't, this is just a summing up. Don't study it from here. Study it from the pages, from the figure of Kubi, where I explain how the positive and the negative selection is actually occurring. This is a summary concept, summarizing concept or a panoptic view of what a T cell tolerance is sent at the central level. And this is the T cell tolerance at the peripheral level. As I said, that even though MTECs, that is the medullary epithelial cells, are capable of presenting almost 90% of the self-antigens of the body, they do fail in expressing the 10% of the self-antigens. As a result of which, some self-antigens escape. So at the peripheral level, if certain self-antigens are seen, a variety of mechanism is there to down-regulate the T lymphocytes. There can be clonal deletion and apoptosis. There can be energy. There can be active suppression. There can be immune deviation. Immune deviation is that a mature CD4 population, as just earlier had said, is usually known as T helper 0 cell. So it differentiates into a helper 1 subtype or a helper 2 subtype depending on the cytokine produced. Suppose a cell has developed into a helper 1 subtype by the withdrawal of the cytokines from its local vicinity and reintroduction of cytokines of the helper 2 subtype, the helper 1 can be immediately changed to a helper type 2 category. So these are certain critical concepts in further T cell tolerance development. So there is immune deviation, there is ignorance, that is neglect by, that's no encounter. There might be active suppression by the immunoregulatory cells. There can be energy, that is receptor signaling proper is not done. Or if there is a hyperactivated T lymphocytes, the T lymphocytes are forced to die by the T regulatory cells. So these are various ways where at the peripheral level, the tolerance can be introduced. So this was a summing up of T cell development. And with this, I come to the end of this topic. With fourth sem, I think so. I have one more topic, that is organs and cells and organs of the immune system. Cells I have done in the class. Still, I'll give a brief recapitulation. I'll be mainly taking up immune organs in the online class.